Hey there, science fans. Today we're diving into the fascinating world of DNA, transcription and translation. These processes are fundamental to life as we know it. They're how our cells use the instructions encoded in our DNA to build the proteins that make us us. Think of DNA as a blueprint. DNA, or deoxyribonucleic acid, is the molecule that carries the genetic instructions for life. It is composed of two strands that coil around each other to form a double helix. Each strand is made up of a sequence of nucleotides, which are the building blocks of DNA. These nucleotides are composed of a sugar, a phosphate group, and a nitrogenous base. The sequence of these bases encodes the genetic information that determines the characteristics of an organism. DNA is found in the nucleus of eukaryotic cells and in the cytoplasm of prokaryotic cells. It is organized into structures called chromosomes, which are passed from one generation to the next during cell division. But how do our cells actually read and use these instructions? That's where transcription and translation come in. Get ready for a wild ride as we explore the amazing molecular machinery that makes life possible. Let's start with transcription, the first step in gene expression. During transcription, a segment of DNA is copied into RNA by the enzyme RNA polymerase. This RNA molecule, called messenger RNA, mRNA, carries the genetic information from the DNA to the ribosome, where it will be translated into a protein. In eukaryotic cells, transcription occurs in the nucleus, and the mRNA must be processed and transported to the cytoplasm before translation can occur. This processing includes the addition of a 5' and a poly A tail, as well as the removal of non-coding sequences called introns. So how do we bridge the gap? With RNA, or ribonucleic acid, is like DNA's messenger. During transcription, a special enzyme called RNA polymerase binds to the DNA at the beginning of a gene. It then moves along the DNA strand, reading the sequence of nucleotides and creating a complementary RNA molecule. This RNA molecule, called messenger RNA, mRNA, carries the genetic instructions from the DNA in the nucleus out to the cytoplasm, where proteins are made. Once the mRNA reaches the ribosome, the process of translation begins. Translation is the process by which the genetic code carried by the mRNA is decoded to produce a specific protein. This process involves the ribosome, transfer RNA, tRNA, and various other factors. The ribosome reads the mRNA sequence in sets of 3 nucleotides called codons. Each codon specifies a particular amino acid which is brought to the ribosome by a tRNA molecule. tRNA molecules act as interpreters, carrying specific amino acids to the ribosome based on the sequence of codons on the mRNA. The ribosome then links these amino acids together to form a polypeptide chain. This polypeptide chain will fold into a functional protein. And now that we have our mRNA transcript and our trusty RNA helpers, it's time for translation. This is where the genetic code gets deciphered and turned into a functional protein. Remember those ribosomes we talked about earlier? Well, this is where they shine. The ribosome attaches to the mRNA and starts reading the codons, those 3 nucleotide sequences that code for specific amino acids. Each tRNA molecule, carrying its corresponding amino acid, binds to the matching codon on the mRNA. As the ribosome moves along the mRNA, it links the amino acids together in the order specified by the codons, forming a polypeptide chain. This chain will eventually fold into a unique 3-dimensional structure, becoming a fully functional protein. This process is essential for the growth, development, and maintenance of all living organisms. See you later in more videos. In this chapter, we will explore the differences between prokaryotic and eukaryotic DNA transcription and translation. Prokaryotic cells, which lack a nucleus, perform transcription and translation almost simultaneously in the cytoplasm, pores 2s. In contrast, eukaryotic cells, which have a defined nucleus, separate these processes both spatially and temporally. Transcription occurs in the nucleus, where the DNA is transcribed into mRNA. The mRNA then travels to the cytoplasm, where translation occurs, pores 2s. This separation allows for more complex regulation and modification of the mRNA before it is translated into proteins. Understanding these differences is crucial for comprehending the complexity of cellular processes in different organisms. Pores 2s. Thank you for joining us on this journey through the microscopic world of DNA, RNA, and proteins. Stay tuned for more videos where we'll delve even deeper into the marvels of biology. See you later in more videos.